Evolution is a change in the heritable characteristics of a population over generational time. For evolution to occur, the characteristics must be heritable. In other words, the characteristics must have a genetic basis, which means that they could be passed on from parents to offspring. The theory of evolution by natural selection was coined by Charles Darwin and is now well understood. However, Charles Darwin's theory was not always the only theory of evolution. Jean-Baptiste Lamarck proposed a theory for evolution based on the inheritance of acquired characteristics. An acquired characteristic is when an organism has a change of function or structure in direct response to the environment. For example, according to Lamarck, the giraffe got its long neck because its ancestors stretched their necks to eat leaves that were just out of reach. This stretching of the neck was passed on to their offspring over generations until it reached its current length. Darwin argued that evolution and variation within a population occur due to selection pressures. This enables organisms that are better adapted to the environment to survive and pass on this advantage to future generations. For example, this population of mice has moved into a new area where the rocks are very dark. Due to genetic variation, some mice are black and some are white. Mice white in color are more visible to predatory birds than black mice. Thus, the white mice are eaten at a higher frequency than black mice. Only the surviving mice reach reproductive age and produce offspring. Because black mice have a higher chance of producing offspring than white mice, the next generation contains a higher proportion of black mice than the previous generation. Darwin's theory was and continues to be supported by many lines of evidence. DNA and proteins provide evidence for evolution. When we compare the DNA sequences of different species, we find remarkable similarities. These similarities provide evidence that all life on Earth shares a common ancestor. For example, humans and chimpanzees share about 98 to 99% of their DNA. The fact that the DNA is so similar helps support that humans and chimpanzees are closely related and share a recent common ancestor. When comparing a part of a mouse gene to the gene of a fly, it can be seen that there are many similarities in one section of the gene. Each highlighted base represents DNA bases that are the same. But it doesn't stop there. DNA and RNA sequences are transcribed and translated into proteins, which are chains of amino acids. The genetic code determines the sequence of amino acids in a protein. Scientists can trace evolutionary relationships by comparing the amino acid sequences of proteins from different species. For instance, the protein cytochrome C can be compared in several species. Cytochrome C is a small protein that plays an important role in the electron transport chain within the mitochondria of all cells. When comparing each of these species, we can see that a human and a monkey only have two differences in the amino acids in cytochrome C. A human, compared to a duck, has 11 differences in the amino acid found in cytochrome C. And a human and yeast both have this protein, but have 51 differences. These trends further support the theory of evolution, as we can see that species develop over time, gradually diverging from a common ancestor as a result of differences in natural selection. Selective breeding also provides evidence of evolution. Did you know more than 300 breeds of dogs are recognized internationally? Did you know one type of plant, the mustard plant, gave rise to many different vegetables we know today, such as broccoli, cabbage, and kale? These differences did not happen by chance. They are a result of selective breeding, a process that provides strong evidence for evolution. Selective breeding, also known as artificial selection, has been used by humans for thousands of years. It all started with our ancestors selectively breeding wolves with desirable traits, eventually giving rise to the many diverse dog breeds we have today. Selective breeding involves choosing parents with specific traits to produce offspring that exhibit those traits. Over many generations, this can lead to significant changes in a species. What kinds of characteristics might have been desirable in dogs? A dog's temperament, such as friendliness, loyalty, or aggression, might have been a trait chosen by humans as suitable for different needs. In addition, a dog's intelligence and ability to learn commands quickly, or their size or speed, could have been traits selected by humans. Another example of selective breeding can be seen in agriculture. Centuries ago, farmers identified characteristics in some wild mustard plants that they wanted to keep and planted seeds from those plants, gradually emphasizing certain traits. Plants with desirable leaves eventually became kale. Plants with large terminal buds were bred to produce cabbage. In the case of broccoli, as well as cauliflower, the flowers were the focus. The considerable changes that have occurred in domesticated animals and crop plants over relatively short periods of time show that artificial selection can cause rapid evolution. Artificial selection achieved this in about 12,000 years, during which humans have been growing crops. It therefore seems reasonable to assume that natural selection could have caused major evolutionary changes over the billions of years of life on Earth. Additional evidence for evolution comes from homologous structures. Homologous structures are body parts in different species that have similar underlying anatomy, even if they serve different functions. Taking a look here, we can see that despite their different uses, the bones share the same basic arrangement: a single upper bone, the humerus, followed by two lower bones, the radius and ulna, and then several smaller bones in the wrist and fingers. These similarities are not coincidental. They indicate that these animals share a relatively recent common ancestor. Over millions of years, natural selection has modified these structures to fit different environments and functions. Homologous structures can be found across many different species, providing a map of evolutionary relationships. These homologous structures result from a process known as divergent evolution, which is when one ancestral species gives rise to new species adapted differently to their different environments. Have you ever noticed how some animals look similar, even though they are not closely related? This phenomenon is called convergent evolution, and it results in what we call analogous structures. Convergent evolution explains why structures have similar functions, but very different origins. For example, the wings of a hummingbird, a bat, and a butterfly all share a similar function, flight, but do not indicate a recent common ancestor. Convergent evolution occurs when different species evolve similar traits independently, often because they live in similar environments or face similar selective pressures in their environments. Take a look at the shark and the dolphin. Both have streamlined bodies and fins to help them swim efficiently. However, sharks are fish and dolphins are mammals. Differences in the structures of their fins support that these similarities arose because they both adapted to life in the water, not because they share a recent common ancestor. Speciation is the process that results in new species. It can result when one pre-existing species splits into two or more new species. When species become different, having been separated from each other for an extended amount of time, they could become new species. Individuals of one species become separated and begin adapting to slightly different conditions. Natural selection will allow for them to develop in two different ways until they become recognizably different. The finches of the Galapagos Islands provide clear examples of how species arise. Scientists believe that a bird species called the grassquit first made its way to the Galapagos Islands over 2 million years ago. The bird flew from mainland South America, possibly Ecuador, and arrived on the small volcanic islands of the Galapagos archipelago. The grassquits migrated to various islands in the Galapagos. When the birds arrived on two different islands, they created two separate populations of the original grassquit species. Each island has distinct and different food sources and habitats. On one island, the birds can feed easily on ground seeds from the abundant plant life, while on the other island, cactuses are the most common plant and food source. Here is where natural selection comes into play. On the first island, a short, stout beak was beneficial for eating seeds from the ground. Birds with this beak type were more likely to survive, mate, and pass the short beak trait on to their offspring. Meanwhile, on the second island, a longer pointed beak was far more useful to pierce cactus fruit and feed. So likewise, this pointy beak trait meant a higher chance of survival and was passed genetically onto their offspring. Now, if we fast forward over hundreds of thousands or more breeding generations, the advantageous beak traits on each island have been passed down to young birds many thousands of times. During this process, the size and shape of bird beaks between the two island populations have become significantly different. The birds on each island are now so different in appearance and behavior that they are recognized as separate species. Not only that, but they are also now significantly different from the original pioneering South American grassquit. The single bird species that arrived in the Galapagos has now evolved into two different and unique species of the Galapagos finch. If we take our example one step further, imagine the grassquits originally arrived on many different Galapagos islands. Now we have many new bird species that have evolved from the original bird. This model shows how the common ancestor from the South American mainland experienced divergent evolution as birds adapted to the various foods available on different islands found throughout the Galapagos. Speciation increases the total number of species on Earth. However, extinction can also occur, and extinction decreases the number of species on Earth. Reproductive isolation and differential selection are two fundamental mechanisms that drive speciation. Reproductive isolation occurs when two populations of the same species are prevented from interbreeding. Isolation can happen due to various factors such as geographic or behavioral isolation. In geographic isolation, populations become physically separated by barriers such as mountains, rivers, or distance, which of course means they are not able to reproduce if they cannot come in contact with one another. Behavioral isolation includes differences in mating or rituals that prevent populations from interbreeding. For example, distinct mating calls or dances can act as barriers. The concept of isolation is based on the concept of gene flow. Gene flow is the introduction of genetic material from one population of a species to another. More gene flow results in two populations being more similar genetically. Less gene flow results in two populations being more different. Speciation is a continuum. When populations are still connected by gene flow, they are less likely to become two distinct species. However, when there is a lack of gene flow between populations, this allows them to begin evolving independently. In addition, differential selection must also occur. Different environmental pressures or selective forces act on those separated populations, which leads to a divergence in traits. This can happen in several ways. Environmental differences might exist if the two populations are geographically isolated and face different environmental conditions, such as temperature or food availability. These differences in selective pressures could lead each population to adapt differently to their specific environment. Another example of differential selection could relate to the types or levels of predation and competition in each population's environment. This could drive the evolution of distinct defensive traits or competitive strategies. Over time, each group that is reproductively isolated and experiencing differential selection must accumulate enough differences genetically that if these two groups were to come into contact again, they might not recognize each other as potential mates or be able to successfully interbreed. At this point, the reproductive barriers are strong enough to prevent interbreeding between the groups, and they are considered separate species. The speciation of bonobos and chimpanzees is a classic example of how geographic isolation and differential selection can lead to the formation of two distinct species. About 1 to 2 million years ago, the Congo River created a significant geographic barrier in Central Africa. The river effectively split the ancestral population of chimpanzees into two groups. The two populations were reproductively isolated. In addition, there was differential selection due to the different environmental conditions on either side of the Congo River. The northern side, home to common chimpanzees, was more prone to seasonal variation and a mix of forest and savanna. The southern side, home to bonobos, was more stable and consisted mainly of dense tropical forests. The southern forest was rich in food resources, which reduced competition for food among bonobos. This abundance allowed for the development of a more peaceful and cooperative social structure. The northern environments had more predators and competitors, leading to the evolution of more aggressive and hierarchical behaviors in common chimpanzees. Over time, the isolated populations accumulated genetic differences due to mutation and natural selection. These genetic changes further reinforce the physical, behavioral, and social differences between bonobos and chimpanzees. After enough time of isolation, even if bonobos and chimpanzees were to come into contact again, the genetic, behavioral, and social differences that have accumulated would prevent successful interbreeding. This reproductive isolation solidifies their status as separate species.